Chapter 12, Part 3. In this chapter, I want to talk about the plasma membrane of the neuron. Neurons contain transport protein for moving substance across the plasma membrane. These include both pumps and various types of channels. Transport proteins were discussed in Chapter 4, and I want to review them for you. In the picture in this slide, you can see the phospholipid bilayer in the cell membrane. And in this bilayer, you can see a scattered transport proteins. Some of them are pumps and some of them are channels. Channels can provide the need for a substance to move down its concentration gradient. Neurons contain the following major types of channels. First, we have leaky channel. Second, we have chemically gated channel. And the third one is called voltage gated channel. The leaky channels or passive channels are always open, allowing continuous diffusion of a specific type of ion from a region of high concentration to a region of low concentration. Examples of leaky channels are sodium-ion leaky channels and potassium-ion leaky channels, and they don't need any ATP to transport any material. The second one is called chemically gated channels. These channels are normally closed. They temporarily open in response to binding of a neurotransmitter. When they become open, they allow a specific type of ion to diffuse across the plasma membrane. Examples of chemically gated channels include chemically gated potassium channels, chemically gated chloride channels. As you see in this picture, we have a transport protein which contains a specific location for combination to the ligand. Our ligand may be a neurotransmitter. A neurotransmitter attached to our protein, the gates become open and it lets the substance to go down its concentration gradient. Again, we don't have energy for the chemically gated channel. The last type of channels is called voltage gated channels. These channels are normally closed, but they become open in response to a change in electrical charge across the plasma membrane. As you see in this picture, out of the membrane is positive and inside is negative. When we have a change in the plasma membrane charge, outside become negative and inside become positive, the channel become open and let the particle to pass. We have voltage-gated sodium channel, voltage-gated potassium channel, and voltage-gated calcium channels in our neurons. In this slide, you can see two different pictures. Picture A talks about chemically gated channel. As you see in this picture, we have a protein. This protein contains a site for attached to the neurotransmitter. When the neurotransmitter attached to this site, the channel become open and it lets sodium ion to come into the cell and potassium ion to go out of the cell. And in picture B, you can see a voltage-gated ion channel. Out of the cell is positive and inside is a negative. When we have a change in the electrical charges on both sides of the membrane, the channel become open and let the sodium ion to pass across the membrane. The next type of protein which we have in the cell membrane is called pumps. Pumps maintain a specific concentration gradient by moving substance against a concentration gradient, a process that requires cellular energy. The plasma membrane of the neuron contains both sodium-potassium pump and calcium pump. A great deal of energy is required to power the vast number of sodium-potassium pumps in a neuron plasma membrane. Approximately, it's about 2/3 of a neuron energy. In this slide, you can see the different states which we have for voltage-gated sodium channel. In the first picture, you can see resting state. Although the inactivation gate is open, the activation gate is closed, and entry of sodium ion is prevented. In the second picture, which is called activation state, both the inactivation gate and the activation gate are open. Sodium ion move into the cell through the open channel. And in the last picture, which we call it inactivation state, the activation gate is open, but the inactivation gate is closed. So the sodium channel becomes closed and sodium ion cannot come into the cell. So we have these three different states for voltage-gated sodium channel: resting, activation, and inactivation state. Only during activation state the sodium ion can move. In resting and in inactivation state, the entry of sodium ion is prevented. In this picture, you can see the distribution of pumps and channels in all parts of our neuron. In the plasma membrane, we can see leaky channel for sodium ion, leaky channel for potassium ion, and sodium-potassium ATPase pump. And all of these three pumps and channels can maintain the resting membrane potential for our neuron. In this picture, you can see the distribution of pumps and channels in the plasma membrane. The first part of our neuron is called receptive segment. The receptive segment contains the cell body of the neuron and the dendrites. In this part, you can see chemically gated cation channel. The cation channel means the channel for movement of sodium ion. You can see chemically gated potassium channel and chemically gated chloride channel. In the next segment of the neuron, which we call it initial segment, and the initial segment is actually the axon hillock, you can see voltage-gated sodium channel and voltage-gated potassium channel. In the next part of our neuron, which is our axon, and we call it conductive segment in neurophysiology, you can see voltage-gated sodium channel and voltage-gated potassium channel. And in the last part of our neuron, or in the synaptic knob, which we call it transmissive segment in the neurophysiology, you can see voltage-gated calcium channel and also calcium pump. These are the distribution of pumps and channels in the plasma membrane of our neuron. Now, answer these questions. Describe the three states of voltage-gated sodium channel in neuron. We have three different states: resting, activation, and inactivation state. Which functional segment of a neuron contains chemically gated channels? What do you think? The chemically gated channels, yes, are located in the receptive segment. Which functional segment contains voltage-gated channels? The axon hillock, the entire part of our axon, and also the synaptic knob. In neurophysiology, we call them initial segment, conductive segment, and transmissive segment. Initiating and transmitting electrical current is central to the function of neuron physiology. Necessary to this process is establishing and maintaining a resting membrane potential that can be changed from its resting value. Now, I want to talk about electrical energy. Electrical energy is the movement of charged particles, and that is all usable form of energy which is available to do work. Neuron activity is dependent upon electrical energy, specifically electrical current. Here we have three different parts. One of them is called voltage, one of them is called current, and the other one is resistance. The voltage is the measure of amount of difference in electrical charge between two areas and represent potential energy. The unit is volt or millivolt. The current is the movement of charged particles across the barrier, which separates them. The greater the movement of charged particles, the greater the current. And the resistance is the opposition to the movement of charged particles. This is the barrier between the charged areas. The larger the resistance, the lower the current. The relationship of voltage, current, and resistance is expressed by Ohm's law, and it says that current is equal to voltage over the resistance. This expression shows that current is directly related to voltage and inversely related to resistance. Thus, a greater current is responsible with a larger voltage difference and a lower resistance. Now, I want to talk about these three items in neurons. Charged particles are ions such as sodium ion and potassium. There is a difference in charge on either side of the plasma membrane due to an unequal distribution of ions. The plasma membrane phospholipid bilayer offers resistance because it generally doesn't allow the passage of ions, and resistance is altered across the plasma membrane through ion channels that open and close. Resistance is decreased when ion channels are open and increased when ion channels are closed. And current is generated when either positively charged ion or negatively charged ion diffuse across the plasma membrane through open channels. The neurons at rest have some characteristics. The ions are unevenly distributed across the plasma membrane due to the action of the pumps. We have higher concentration of potassium ion in cytosol when we compare it with interstitial fluid. We have higher concentration of sodium ion, chloride, and calcium in interstitial fluid than in cytosol. Gated channels are closed in the functional segment of the cell, and there is an electrical charge difference across the membrane. Cytosol is relatively negative when we compare it with interstitial fluid. Resting membrane potential is typically negative 70 millivolt, and this difference can be measured with microelectrodes and voltmeter, which I will show you in the next slides. In this slide, you can see the phospholipid bilayer of a cell in orange color. The blue color shows you the transport protein in the cell membrane. And you can see the amount of sodium ion and chloride is more out of the cell. The amount of potassium ion and organic anions or the proteins are more inside the cell. So, out of the cell is positive and inside the cell is negative. And the difference between outside and inside is negative 70 millivolt. This is called resting membrane potential or RMP. In this slide, you can see a neuron at rest. At rest, a neuron has a negative resting membrane potential, which on average is negative 70 millivolt. All gated channels are closed, and concentration gradient exists for sodium, potassium, and chloride along the length of the neuron, and for calcium at the synaptic knob. Again, we have a plasma membrane of the neuron at rest during resting membrane potential. As you see in this picture, RMP or resting membrane potential is established and maintained by potassium leak channels, sodium leak channels, and sodium-potassium pump. And you see that we have the microelectrodes inside and outside of the cell membrane. And the difference between outside and inside is negative 70 millivolts, which is showed by our voltmeter. For review of the resting membrane potential, you need to know that the potassium diffusion is the most important factor in setting resting membrane potential. Potassium diffuses out of the cell due to its concentration gradient. Potassium diffusion out is limited by the electrical gradient. If potassium were the only ion that leaked, resting membrane potential would be where the potassium concentration and electrical gradients are at the equilibrium, which is negative 90 millivolts. Since there are a few sodium leak channels, sodium also influences the resting membrane potential. Sodium diffuses into the cell due to its concentration gradient. It comes down its concentration gradient from outside to inside. And these small sodium leaky channels can make the resting membrane potential less negative. So from negative 90, it becomes negative 70 millivolts. The role of sodium-potassium pump is important for our neuron. This pump can push 3 positive charges out of the cell and 2 potassium or 2 positive charges into the cell. And it only contributes about negative 3 millivolts out of negative 70 millivolt total. So this pump is very important for maintaining the concentration gradient of these ions on both sides of the membrane. I will post these questions on the assignment folder in your Blackboard, and you're supposed to answer these questions for me there. Here, I want to discuss the physiologic events that occur in the functional neuron segment from the time of initial stimulation at the dendrite and the cell body until release of neurotransmitter from the synaptic node. An overview of these events that occur in the receptive, initial, conductive, and transmissive segment is provided in this picture. As you see in this picture, the blue part in the beginning of the neuron is called receptive segment. The receptive segment is the cell body and the dendrites. Here we have binding of neurotransmitter and generation of graded potential, which I will talk about it in the next slide for you. The next part of our neuron is called initial segment. In the initial segment, which is actually the axon hillock, all the graded potential, all the stimulation which come to our neuron are added with each other, and it can make the action potential for us. The next part of our neuron is called conductive segment. The conductive segment is the length of our axon, and in this length we have propagation of action potential. And the purple part of this slide shows you transmissive segment, which is actually the I want to begin my discussion on this picture, which shows you the arrangement of several perisynaptic neurons in close proximity to one postsynaptic neuron. Between each perisynaptic and postsynaptic, we have a fluid-filled synaptic cleft. Each perisynaptic neuron releases neurotransmitter which binds to the receptive segment of postsynaptic neuron. The membrane potential that act as signals. We have two different types of signals. One type of signal is called graded potential. The graded potential are a small, short-lived change which we have in the receptive segment of our neuron. And the other type of potential is called action potential, which is the long distance signal which propagates in the length of the axon. First, I want to discuss the graded potential for you. The graded potential are small, short-lived changes in the resting membrane potential with the following characteristic. First, they are established in the receptive segment by the opening of chemically gated ion channel. It's very important to know only chemically gated channels are important here. They make some local changes which are very short-lived. They vary in degree of change and direction of change of the resting membrane potential. Some of them are very small, some of them are large. They can make depolarization or they can make hyperpolarization of the cell membrane for us.
In this picture, you can see multiple perisynaptic neurons associated with one postsynaptic neuron. Some of these perisynaptic can make EPSP and some of them can make IPSP, as you see by positive and negative sign. The degree of change in the resting membrane potential is dependent upon the amount of neurotransmitter bound per unit of time. More neurotransmitter make more channels become open and we have greater change in the membrane potential.